Hi everyone, in this video we will be talking about mitosis. We will be explaining about the importance of mitosis, the process of mitosis, and the abnormalities of mitosis.
For the first part, we're going to see about importance of mitosis. Mitosis is a process of cell division which plays a key role in growth, cell replacement, and also sexual reproduction. As for growth, mitosis ensures that increase in number of cells in tissue and organs which is necessary for overall growth and development in an organism. Moreover, mitosis replaces worn out or damaged cell, ensuring that the tissue functions and also remains its structure by producing new identical cells. Lastly, mitosis is the mechanism which organisms reproduce sexually and creating offspring that genetically identical to the parent. So as it produces two daughter cells with same DNA as the parent.
Another importance of mitosis is genetic stability. Mitosis makes sure all the new cells are genetically identical to the original or parent cell. This means the same number of chromosomes and the same genetic information is passed down. Next is regeneration. Some animals can grow back missing body parts because of mitosis. When cells divide through mitosis, they regenerate new cells, replacing old or damaged ones.
Next, we will be talking about the process of mitosis. Let's start with prophase. During prophase, the chromatins in the nucleus begins to condense into thick strands. Each chromosome consists of two chromatids, which are known as sister chromatids. They are joined at the centromere. The nucleus disappears and the mitotic spindle begins to form. The mitotic spindle will be completed at the end of the prophase. Centromeres move apart to opposite poles of the cell and is being pushed by the mitotic spindles between them.
Now let's talk about what happens in the metaphase. First, the centrosomes move to opposite poles of the cell. Next, the mitotic spindle forms, stretching across the cell. The chromosomes line up in the middle of the cell. This middle line is called the metaphase plate. Each chromosome is ready to split in half, with its centromeres holding it in place.
In anaphase, paired centromes of each chromosome separates into two chromatids are now separate from each other. After separation, each is considered a full chromosome, which is daughter chromosome. After that, contraction of spindle fibers pull the sister chromatids apart towards the opposite poles of the cell. The process after this is called telophase, which we will see in the next slide.
Now for this final stage is telophase. So the spindle fiber disappears and chromosomes begins to uncoil and return to its chromatin form. The nucleus starts to form at each pole and new nuclear envelope reforms around each set of chromosomes. The nucleolus reappear in both nuclei and signaling the completion of nuclear division. At this point, the division of the nucleus into two genetically identical nuclei is complete. Finally, cytokinesis begins and marking the start of the process will divide the cytoplasm and complete the cell division.
Last but not least, let's talk about the abnormalities of mitosis. Firstly, let's talk about malignant tumors. Malignant tumors, or known as cancer, is formed when mitosis occurs uncontrollably. Cancer cells spread and destroy normal cells, which affects the functions of tissues. Malignant tumors are often caused by radiation, such as X-rays and gamma rays, carcinogens, bacteria and viruses, and also genetic factors. Next is benign tumors.
Abnormalities in mitosis, such as error in chromosome segregation, failure of cell cycle checkpoints, or gene mutation, can lead to uncontrolled cell proliferation, resulting in benign tumors. These tumors are non-cancerous and typically grow slowly, often without spreading or invading surrounding tissues. Although they may cause problems by compressing nearby structures, they remain localized due to the lack of malignant behavior, unlike cancerous tumors.
That's all from us. Thanks for watching.